This is the first half of Chapter 50. We're going to do this in two parts because it's such a major chapter with lots of concepts in it. The first half we're going to do is going to be our sensory mechanisms, and the second half is going to be your response to the senses, which is muscle movement. A key concept to remember throughout the first part of this chapter is that all of the senses are coming from the idea of kinetic energy. Some form of kinetic energy or chemicals, be it sound, heat, light, or physical impact. All of these are kinetic energies that your body needs to be able to sense. Plus, we'll add that chemical scenario with the chemical reception. Now, your nervous system doesn't work off of kinetic energies and carrying sound somehow deep into the recesses of the brain and making that into some kind of understanding. Your brain works off action potentials. Your entire nervous system, as we learned in Chapter 48, was action potential, and for good measure, some more action potentials. And that's it. It's ones and zeros. It's binary. So how do we convert the idea of some form of kinetic energy, like, for example, pressure? How do you convert that into the language of action potentials? Once we've done that, and the brains make a decision about that, the second half of this chapter that we'll cover in the next lecture is muscle motion, because usually the only reason you have a stimulus that you can sense, the only reason you have a sensor, is so you can respond to it. And in we animals, that response is, of course, movement or some kind of glandular secretion or something like that. There's a lot of sensors that we're not going to talk about, such as the ones in the endocrine system. We won't specifically discuss the sensors that are involved, for example, in your pancreas, that are sensing blood sugar, and based on them, you know, your beta islets secrete insulin, or if the alpha islets sense it dropping too low, they'll secrete glucagon. That's still a chemical receptor, but it's more a direct contact that involves a little less of the nervous system. Okay, so here's the basic principle. First, you have a sensory input. Okay, they're like, hey, there's a worm. I want to eat the worm. Then the brain has to integrate that and think about that and translate what that means. To you, that may seem automatic, and to us, it's somewhat, ethereal in nature, the higher levels at which we think, because we go so far beyond, is the food present? We go into, is the beef particularly well seasoned today, or shall I have the chicken? Our decisions are very much more integrated than your wild animals' decisions are. But in your basic sense, you have a sensory input, and then a computer chip called the brain has to integrate that sensory input and spit out a response. Either bite onto something and eat it, or crawl past it because it's a stick, not food. And even though you say, well, duh, it's food, of course it's going to eat it. You got to think to yourself, how is it exactly that a system of action potentials and more action potentials circling around each other in this complex thing called the brain, how did those action potentials eventually spit out an action potential that was movement of the proper muscles to bite a worm? Because that's what we're talking about here. A sensory receptor smelling this food, transforming that information, that chemical information, into an action potential. Those action potentials hitting a brain, the brain responding with more action potentials that say, move these paws forward this many times and then bite. Okay, that's actually fairly fascinating when you think about it. And even further, the fact that I just told you to think about something and you could think about it. Remember, right now you're transmitting the information of sound waves coming out of your computer or your phone. It's hitting your ear. We're translating that into action potentials that are going into your brain and being stuck in another thing called memory. We won't be talking about that, but hopefully that's going to be operating throughout this entire time as well, which is a lame way of saying you should remember what I say. All right, let's talk about sensory receptors. Okay. The first concept is transduction, and this is just like in Chapter 7 when we were talking about signal transduction way back in 2107. In Chapter 7, when we were talking about converting a chemical signal outside of a cell into some kind of message inside the cell, that was signal transduction at a cellular level. Here we're doing sensory transduction, where we're converting some kind of stimulus, be it a form of kinetic energy or a chemical. We're engaging receptors, or we are changing the shape of molecules in some way that we can then change the membrane potential of the sensory receptor. And so we're moving it towards hyperpolarization or depolarization just as before. But this is receptor potential. This may not be a neuron. there might not be such a thing as threshold. This thing might not have an axon coming out of it. This might just be when you change the receptor potential, does it put out more or less neurotransmitter? That's it. Or this receptor could itself be a neuron, and if you give it enough of these receptors being transduced into some kind of change in membrane potential, it might hit threshold and give an action potential directly. But the receptor potential, this is kind of like postsynaptic membrane potential. Coming down the synapse to presynapse is action potential, action potential, action potential. But after the synapse on the receiving cell, it's still a graded potential. We don't know how far it can go. Is it going to go past threshold or not? Is it going hyperpolarized? We're still in the land of receptor potential where we can do graded stimulus. It's once you hit a neuron and you hit action potential that there is no longer any question. You have hit a point, you get an action potential, we're done. Okay, now, two ways a receptor can look. Okay. The receptor can be itself a neuron. Okay. Or the receptor could basically synapse with a neuron. Okay, the first example that I talked about was this receptor that synapses, where it's receiving a stimulus, and based on that stimulus it releases more or less of this neurotransmitter. That neurotransmitter changes the potential of this postsynaptic neuron. Okay, and then if you get enough, it goes to action potential. If you don't get enough, it doesn't. Or we could be talking about the stimulus hitting a neuron itself and it not synapsing at all at the beginning. It just sends a message down, an axon, bam, there you go. The signal's already started. Now, of course, it will synapse further down, but we didn't jump from a receptor cell to a nervous cell. We did both in one package. So if the receptor is a neuron, if you get more receptor potential, what happens is this means more often you'll send an action potential. Very often these neurons are constantly signaling. They're signaling it like once a second, even if there's nothing there to sense, like your taste buds might be signaling constantly, hey, I'm here. But when they engage the right chemical, suddenly they quadruple or quintuple the speed of those action potentials. So that's one way in which your brain can receive the information. It's saying, yeah, I know you're there. Yes, I know you still have a tongue. You're still a taste bud. I get it. But then suddenly if you start hammering those action potentials really fast, it goes, oh, was I supposed to pay attention to you? If the receptor is not a neuron, instead of having to directly alter its speed of action potentials, it could just be releasing more neurotransmitter, which will result in the following afferent neuron changing its frequency of action potentials. But whether or not the receptor is a neuron, or whether or not the receptor synapses with a neuron, the net effect is we speed up or slow down the action potentials. And that's one more thing I want you to know. Every case we are talking about today, until we get to a very specific example, the signal that says, hey, I have found the thing I'm looking for, it could be to release more neurotransmitter, but it could also be to release less. And the effect of that neurotransmitter could be to cause more action potentials, or it could be to cause less. When we get to sight, we're going to find out it works backwards from the way you think it does. All of the receptors in the eyes are sending forth chemical signals to the neurons behind them, and sending action potentials like you wouldn't believe constantly through the rods, the black-white sensors in your eyes, the outlines of objects. They're constantly signaling. It's when light strikes them that they shut up and they stop signaling, and you stop getting the action potential. So suddenly you see light when there's you only get the impression of light when your eyes shut up. Isn't that weird? It's kind of backwards. Okay. So just remember, everything can go up or down. And up doesn't mean I see light or I taste sugar. Up just means up. It's how your brain interprets that information that really makes the difference. Now, what are some ways that we can do this thing that I was talking about? Well, if you do a gentle pressure on the sensory neuron, it will only slightly change its receptor potential, so you'll get a slow pulse, maybe, of action potentials. Let's assume that engaging the receptor depolarizes this neuron. Okay. Well, you could put harder pressure on there, and more of these receptors will open up, causing a faster depolarization, and then it will shut up, and then it will repolarize, and then you open them again so that it goes, so it gets a faster frequency coming through. And you remember from chapter 48, down here, we would expect to see at the next synapse, we would expect to see some temporal summation, where back-to-back action potentials will drive the next neuron over threshold, whereas these action potentials up here coming slow in a repeating pattern weren't frequent enough. They would bump it up, but then it would come back down. They bump it up, towards depolarization, it would come back down. But when you hit it, bump, 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 you caught it faster than the neuron could reset itself and eventually shove it over threshold. Now, another way that your brain can interact with this information is not just necessarily frequency, but it could be numbers. It could be numerical. Gentle pressure and strong pressure might get the exact same number of pulses. It just might be that no pressure gives you this frequency, and any pressure gives you a rapid frequency. But your body can interpret it as light versus hard pressures, not based on the number of action potentials, but based on the number of receptors that all got the same message. Okay, or it could be the depth of the receptor. Instead of just having a receptor, all these receptors right at the surface, it might have a line at the surface and a line down beneath. And so you count it as gentle pressure when the ones up top are engaged, but when the ones down bottom start talking, suddenly your brain says, ooh, you had to push harder to get down to them. I will consider this a hard pressure. Or it could just be, hey, a whole range of these got hit all at the same time. I doubt that was a light touch. It was probably a heavy touch. Okay, so these are the ways your brain has to determine what the information is like. And that brings us to the concept of perception. There is no such thing as sight. Okay, we are all blind. We think we can see, but we don't. Sight is an illusion created by your brain. Okay. Light doesn't have these, I'm not trying to destroy your aesthetic sensibilities here, but light does not have these pretty colors and these gorgeous images that we think it has. It has wavelengths. It is kinetic energy. It has wavelengths. It has frequencies with which it bounces up and down. And those frequencies that bounce up and down engage different receptors in your eye based on the pattern of where they came from and off the angle that they came from. And as they engage those receptors in a repeating pattern over and over, because it hits them and then the next millisecond it hits them again, because light tends to kind of constantly come off that. For example, I'm looking at a brown chair. It's constantly reflecting light at my eye. My eye has seen over the past few seconds thousands of times it's registered that brown light. That mix of wavelengths that I consider to be brown. But see, you know what? That brings it up. There's no wavelength called brown. Brown is just an impression that your brain creates based off the fact that there is red and yellow and some orange and a little bit of green coming off of that chair. And because of that, my brain says, I will consider that to be brown. But there's no such thing as brown light. So my brain is creating this mixed color that does not exist in the rainbow. You cannot, pick a color out of the rainbow called brown. There is no wavelength like that. Okay, there's no wavelength called black. How is it that I look at the computer in front of me and see black? Black is not a color. It's the absence of color. That computer's not dark. It's not that computer has no light coming from it. I can tell there's some light reflecting, but my brain has interpreted it to be black. Okay, the key to this, and the reason why I'm trying to ruin all your fantasy, all your illusions about sight, is that all you have coming from your eyes is a pattern of ones and zeros, action potentials, on, off, on, off, on, off. And all those action potentials are identical, and they are identical to the action potentials coming from your tongue when you eat a steak, which are identical to the action potentials coming from your finger when you stick it under hot water. The signal coming to your brain is identical. In every case, it's always an action potential. But how does your brain create a perception? It's because your brain knows where each receptor is and what type of receptor it is. They have independent wiring going up. That's part of the reason the spinal cord is so thick. Okay, as every receptor kind of has its own signal that it sends up. They don't share. So the moment that action potential from my finger gets to my brain knows that those receptors that are signaling are temperature receptors, that the receptors for hot, not for cold, and that they are in the tip of my left index finger. So when I put it under the hot water, my brain interprets it and gives me the perception of heat in my left index finger. But if you could trace those wires and find them and sort them out, you could squirt a little bit of chemical in there, generate an action potential, and I would tell you that my finger is hot. My left index finger is hot, even though you're doing nothing to it. Okay, so come back to this philosophy that the brain, there is no such thing as tasting sugar. As far as your brain is concerned, when it comes down to it, what it is your brain sensed action potentials, and it knows those action potentials are connected to your tongue, to a chemical receptor for sugar. And therefore it tells you, you just tasted sugar. But all that your brain received was action potential. Let's talk about these receptors, because to me those are the big key. Now, the action potentials we've already talked about, but how do you get it? Okay. Well, let me just jump into them. Mechanicoreceptors are ones that are actually, to me, the hardest to think of. How is it that you convert? I mean, like a chemical sense? Come on, that's easy. A chemical can just bind to the receptor on the surface of a cell and a signal transduction pathway starts. That's easy. But mechanical? Okay, well, there are scenarios here like a hair movement. What you do is you put some dendrites coming off of a neuron, okay, up around a hair cell, and as you flick the hair cell one way, it stretches the dendrites, and maybe these two were making a connection with each other and now they are separated, or maybe they were separated but they were shoved together when you moved the hair. Something about the movement torques the plasma membrane to make or break a contact, and suddenly that's going to open some channels. So touching the two membranes together that didn't used to touch could put a signaling molecule on one membrane in connection with a receptor on the other edge of the membrane, and now they're touching each other, they've been engaged, and we open a channel. Okay, and we let in some salt. Bam, there you go. That's how mechanical force can do that. The same with pressure. Okay, sensors, movement sensors. They usually have a cushion around them. Okay, a big ball of fluid, and when you press on it, it squeezes the plasma membrane, it squeezes those dendrites, and physically, sometimes it actually physically torques proteins open, channel proteins that cause ions to flow in. These are examples of that. Okay, strong pressure sensors tend to be both deeper and have more layers of padding, so that it's harder to trigger them. So if you gently tickle the surface of your hand, that gives a different impression than if you start poking it real hard. It's a completely different feeling, not because the signal's any different. It's just an action potential. But your brain, in the first case, sensed only messages coming from the surface receptors, the ones that are easy to excite. Those strong pressure ones aren't saying anything's happening. But when you suddenly start striking the strong pressure ones, the surface ones sense it. But your brain ignores them because it's more concerned with the fact that, these guys are talking? Well, we don't care about these slight, delicate nuances about the touch, because the touch is hard. So I don't care if it's a tickling versus a scratching touch. What I care about is it's a hard touch. And so it will ignore the surface receptors and go into these. Another mechanical receptor, a very common thing to do, is with that hair. Not only is it the motion of the hair, but it's also the direction that the hair is torqued. Based on how you arrange the dendrites around the hair cell, you can tell if it's being flicked left versus flicked right. And just as I said before, most of the receptors are constantly signaling their neurons, sending out a certain amount of neurotransmitter that's hitting the neuron and giving you some action potentials. Okay, well, if the hairs are shifted right, that changes the receptor potential. Maybe it depolarizes it. Okay, because it opens some sodium channels. Well, now the result of that is not an action potential, because this is not a neuron. The result is, because it's depolarized some, it spits out more neurotransmitter. Okay, but then you flick it the other way and it closes off all of the sodium leak channels, and now we're hyperpolarized, so we send out less neurotransmitter. Now that has an effect on the frequency of action potentials in the afferent neuron, the one coming afterwards, kind of what I call the postsynaptic one. Okay, if you send out more neurotransmitter, maybe that gives you more action potentials. And when you send less, that gives you less action potentials. It's just as possible, though, that these two action potential frames could have been swapped with each other. And that more neurotransmitter would actually slow down the action potentials, right? Because maybe what the neurotransmitter did was it hyperpolarized the cell. Neurotransmitters don't always lead to an action potential. Sometimes they inhibit an action potential. So always remember, it could be excitatory, it could be inhibitory. Don't assume. All right, chemical receptors. Chemical receptors are pretty straightforward. They are basically what we did in Chapter 7. Only instead of just thinking of the receptor on the cell surface, think also that some signal transduction pathway results in calcium release. Because you know calcium release is a great second messenger, and calcium release works on neurons as well, and calcium release tends to cause vesicles to fuse. And those vesicles will dump neurotransmitter. Hey, there you go. Okay, a mechanical process caused calcium to be released, causing neurotransmitter to dump up here, but now it could be a chemical process, just a simple cell signaling molecule. Okay, that's one way in which this could be going on. The book gives you very fancy ways in which this could be occurring, but I want you to think your nose, your tongue, your blood pH, for example, though that's not really the kinds of senses that we're talking about. Blood pH information is still sent to your brain, which tells you how often to breathe. Remember, because we sense the pH of the blood and infer from that how much carbon dioxide is in there, and infer from that that there's the opposite amount of oxygen. That if there's lots of carbon dioxide, there's very little oxygen, and we need to take another breath. That's how that game is played, and this game works through chemical receptors for pH. Okay, and chemical receptors aren't hard. I mean, a pH receptor is really easy. Just have a protein that denatures at acidic pH, and bam, now you can sense acidity because of a protein change. The book also got very fancy with electromagnetic receptors, but these I want you to think of eyes. Okay, I don't want you to think of the beluga whales piloting their way through the sea or thermal imaging from snakes. I want you to think of eyes. Okay, these are the ability to somehow catch light and change it into a physical shape change, which is not hard. You remember in photosynthesis, the whole goal of chlorophyll was to be struck by light, catch the energy, and then change shape based on that. And it started, the chlorophyll in the case of photosynthesis was too overcharged with energy. Electrons jumped to too high of an orbital, so it kicked off an electron. And that's how you did photosynthesis. Similar scenarios happen in your eyes, which is why it's so important for you to eat multiple colored vegetables, because the pigments that it uses for photosynthesis, like beta carotene, you turn around and use those in your eyeballs for sensing light. Only instead of kicking electrons with the pigment, your shape change pigment is going to be wrapped in a protein, and when the pigment changes shape, it changes the shape of your protein, causing it to activate. Thermoreceptors, I mean, this is really easy once again. Just have a protein that changes shape when you heat it or when you cool it. And bam, now you have detected a change in temperature, energy, and transformed that into some kind of chemistry thing, a change in tertiary structure of a protein. Okay, now I'm going to call foul pain receptors, nociceptors. You have to know about them, but they don't belong up here, because all pain receptors, with the one exception of the naked dendrites that just sit in your that just sit out there waiting to be cut, these slice pain sensors. Okay, all other pain sensors are just some other kind of sensor. So the ones that pick up the chemicals from damaged inflamed tissue, that's just a chemoreceptor. That's just a chemoreceptor. That's not a new class. The ones that do excess heat, that's just a thermoreceptor that's buried deeper or got insulation around it. So it's not really its own class. I mean, you think about it, but remember, pain is just an interpretation from your brain. Because one thing hot water can be painful, or it can feel really good, right? Your brain can decide to interpret that hot water as a good sensation. Deep pressure being punched in the arm is interpreted as pain until the guy punching you in the arm is a professional masseuse. And then suddenly you interpret it temporarily as pain, but you also interpret it as pleasure at the same time, all right? Spicy foods hurt until you decide that you like that feeling, and then suddenly your brain craves them. So, it's people talk about these mythical abilities to resist pain and people's abilities to ignore certain conditions. It's not mythical at all. It's a choice. It's a training of the brain to ignore things. An example that I used to use when I was teaching in karate classes, I would tell students, I would say, if you're fighting a person, you're fighting an attacker, a kick to the crotch is a great attack, unless you think the person's a football player. Then don't even try it. Gouge an eye immediately or break a bone, because kick to the crotch, he's going to ignore it for the next 30 seconds because it's just pain. Okay, it's not actually incapacitating him in any way. It's just pain. And if a person is trained to ignore a certain type of pain, then there's no physical quantity to it. It's just action potentials, and it has no effect. Okay, so that's a little bit of application and philosophy there. That's why, you know what? You know how some people can stretch easier and are more limber? Yes, their muscles are more limber, and yes, they don't tear them as often. But also, they've taught their brain to ignore the pain of stretching. The brain said, "Oh, we've stretched this muscle too far," and you said, "No, I haven't." Ignore this, brain, and eventually, brain said, "Okay." Kind of like the fact that your brain is ignoring the existence of the shirt you're wearing. Oops! Until I reminded you of it. Now you can feel your shirt. Is it itching? Can you feel it now? It's touching you all over. You were ignoring it just a second ago. It's proving my point. Okay, now it's time to dig into how some of these receptors translate into senses in a specific sense. So let's start with the sense of hearing. Okay, now the anatomy of the ear I'm not going to walk you through, but we're going to focus on the idea that your eardrum is nothing more than a way to catch sound and to vibrate. So the eardrum vibrates at intensities, so strength of press, and a frequency, so number of wavelengths per second, number of beats per second. And the only purpose of that is to transmit the information from this big fat old ear, all the sound waves it's catching, to this smaller area of the eardrum, which then has this intricate series of bones whose only purpose is then to take that information catching on that bigger area and press it down on a much smaller area. So it's the idea, they really call it it's the anvil method, where you're striking down, some of these bones are called the hammer and the anvil, when we're not calling them incus and stapes, because the idea is, slamming down and putting all the pressure of this gigantic ear that's catching all this sound down on this tiny little oval window. And then those waves are no longer in air, now they're in water. Your body works better with water anyway, so these are actually pressure waves of water coming through. Okay, and now we move into the cochlea, and we move into the auditory area of the cochlea. Okay, so inside this portion, you have what's called the basilar membrane, you have a tectorial membrane, and you have hair cells coming up from the basilar smacking into the tectorial. And what's going to happen is, and here's an example of what they look like. What's going to happen is the basilar membrane is going to vibrate as fluid comes through as fluid waves come through the cochlea. It's going to vibrate based on the frequency of those fluid waves. So it's not like big waves in the ocean. We're talking about rapid, like hundreds of beats per second, sometimes thousands. Okay, so it's really rapid frequencies of pressure waves coming through. And at certain places, this basilar membrane is going to vibrate in response to those pressure waves. The waves will come through in what's called a resonance frequency and cause the basilar membrane to jump up and down. As it does that, the hair cells are smacked against the tectorial membrane, which does not move. The tectorial membrane sits still. Okay, and when these hair cells are pressed up in there, it deflects the hairs sideways and causes the same kind of motion as if we had just done this. Okay, the hair cells get deflected. It doesn't matter that the hair cells weren't the things moving, that it was the cell beneath them that was moving. Your body used the idea of moving hair cells and moving the hairs on them, but instead moving the cell, smashing it upward into something so that forces the hair to bend. So there's your signal transduction step. Now we've taken mechanical energy of vibrating air, turned it into vibrating water, and turned that into hair cells getting slammed up into a tectorial membrane, and now our receptor can change potential because the hair cells are deflected, and that can send either a synapse on an axon, or the sensor itself could be attached to a neuron. Okay, these are all just words that say what I just said. That resonance frequency, though, is an interesting thing. So you don't just hear sound, you hear different pitches of sound and different colors, dare I say, of sound. Sound has texture to you and depth. Now, all your ear is doing is sitting on action potentials, but once again, your brain knows that each hair cell is in a different location along that basilar membrane. Okay, and it turns out that I said resonance frequency. If you it's easier for men to do than women because of the pitch of our voices, but if you go into a stall, a pretty tall stall in a bathroom, and hum a fairly low note, I don't know if my microphone will pick this up, but if you start kind of below that pitch and start working your way up and just slowly going up, it'll sound like this. You'll go and where it got really loud there, you didn't get louder. What happened was you hit the resonance frequency of the room you were in, such that the waves of sound did not just bounce off, they multiplied as they did and caused the room to move with it. So the room started making the same sounds that you were making. This is very similar to blowing air across the top of a Coke bottle. Any kind of soda bottle, you blow across, it makes this woo sound. Drink more of the soda, and the sound goes woo. It gets deeper because you change the resonance frequency, and the bottle is literally vibrating back at you. Okay, well, that's the key of the basilar membrane. So when sound of a certain wavelength comes through, let's say it's a mid-range sound, let's say it's 1,000 hertz. Okay, it doesn't really vibrate point A very much. That's not the vibrational frequency of this portion of the basilar membrane. But when it gets to point B, it's hit the resonance frequency, and this thing bounces up and down at 1,000 hertz at the same frequency of the sound. Okay, but that's not what your brain senses. All your brain knows is that at this point, if these hair sensors get bounced, that means you have a medium tone coming through. Whereas if it's point C, okay, out at the tip, it's got a very low resonance frequency, a much lower number of hertz, lower vibrations or lower sounds. And so it gets the same thing. It gets action potentials, same action potentials it was getting before when you were exciting point B. Okay, but now it knows those action potentials are coming from further out the basilar membrane. So it knows to project that into your brain and into your consciousness as a perception of a deeper sound. Just like sight, there's no such thing as hearing. All there is vibrations. Okay, you interpret lower vibrations as bass wub wub in your dubstep music because your brain told you that that's what it, quote, sounds like. But there's no such thing as a deep sound. There is only the frequency at which it's vibrating. Your brain interprets it to this illusion of sound. Okay, I'm not going over equilibrium. But you can look at that. It's just the same concept of hair cells sticking up, but now we've put this dome over top of them. And if water pushes them sideways, in your ear and balance works the same way that the lateral line system works in fish. And it detects motion, changes in water flowing left versus flowing right. Okay, it deflects the hair cells different ways. And you saw up above, or much earlier on in mechanical receptors, that left versus right gives two different signals that can be interpreted, either faster action potential or slower action potential. Okay, and so that's an example of how we do it. And in fish, what they have is they have basically a balance system running their whole body, not sensing which way they're moving, but sensing which way the water's moving, letting them know if something way back here that they can't quite see moved. They didn't see it move. They felt the pressure waves in the water, and that's why a fish will dart away from you all of a sudden if you move your finger. It felt the water move. All right, now let's talk about light. Okay, whole bunch of interesting things about the eye. Most of this we are going to leave for you to study in lab. So let's instead get some basic parts of the eye down. Okay, your eye actually has multiple lenses. Okay, the cornea acts as a lens. It's just not a mobile lens, it's an immobile lens. Okay, so parts of the eye, you have the outer covering of the eye called the sclera. Okay, this is the white of your eye. The part where that white goes clear is called the cornea. It's the same as the sclera, only it's clear so light can get through it. You have an iris, which opens and closes to control the amount of light coming through, very similar to the diaphragm on the microscope you've been playing with in 2107 and 2108. The pupil is non-existent. Yes, you heard me correctly. The pupil does not exist. It's not an organ. I cannot transplant your pupil. Okay, I can transplant an iris. An iris is an organ, but a pupil is nothing more than a hole. If I dig a hole in the ground, I cannot reach in and pick up the hole. Right? A hole is an absence, and the pupil is just a hole. So that's just a little bit of philosophy I like to tell you all. Some things that we pretend are organs are the absence of an organ. They're a hole. Okay, so that's what the pupil is, the hole to let light in, and it gets bigger and smaller because of this iris. Okay, if you're in too much light, it's going to tighten down so that your receptors aren't getting flooded. The lens is going to bend light. It's how you focus things. And when you try to strain your eyes to change focus, what you're doing is you are making this lens rounder or flatter. The rounder it is, the more sharply it bends the light, and it becomes round when you want to see things that are close up. When you want to see things that are far away, you don't want to bend the light at all. It's coming in at parallel angles. You just want to see it as is, so you flatten out the lens. You've got a retina back here. which is where all the pigmented layer that is catching, all the photoreceptors are there. Interesting, funny, weird thing. The retina has the nerves in front of the photoreceptors. You would expect these nerves to be behind the photoreceptors, but really the nerves, the light's coming in from the left to the right here and striking this as passing through the nerve cells as it does so. You might be able to see better if this weren't the case. And so then that's how you're going to receive the light. Interesting things, you have a hole in your vision. You don't know about it, but if you close one eye, there's a spot that kind of has this feeling. It's somewhere kind of like if you, it's a little to the outside of center. Okay, and there's a trick that I may show you in lecture where you can move your finger back and forth. Focus on one finger, move another until the tip of your finger disappears and you realize you can't see anything in that spot. And your brain is trying to create an illusion of what it thinks the rest of the finger looks like, but it's inaccurate. Okay, that's your optic disc. That's the spot where all those optic nerves come out of the retina and penetrate down into the true optic nerve. Okay, and then you have the fovea. The fovea is the center point that has the most of the cones, the things that detect color. So when you're trying to look at something and tell its fine details and its fine colors, you look straight at it, and when you're looking straight at it, the image of that one object is being projected right there on the fovea. Okay, but if you're trying to see motion in a low light area, you don't want a whole bunch of cones to see color. You want a whole bunch of rods to detect changes in light and dark. And that's why if you're at night, you're trying to spot motion, you don't look directly at where you think the motion is because you're only focusing on its color now. You want to look off to the side so the light strikes you down here or over there, where it's hitting a whole bunch of rods which are focused on the idea of peripheral motion. You know how you go, what was that? And your eye caught some motion, but when you look straight at the motion, it takes you a second to see it moving still, even though your eye saw it in a fraction of a second off to the side. Okay, that's basic eye anatomy. Let's get into how these rods and cones do their job. In rods, what you have is you basically have something similar to thylakoids. You have these little discs. And in these discs, what you have is you have rhodopsin. It's a combination of a pigment called retinol and a protein called opsin. And when light strikes retinol, it changes the shape of retinol. And that change of shape in retinol changes the shape of this protein, causing it to do some stuff, start a signaling cascade. Okay, what's the signaling cascade? Okay, well, suddenly the active rhodopsin activates another signaling molecule called transducin, which grabs hold of phosphodiester ACE. Now, cyclic AMP and cyclic GMP, it's the same thing, it's just with a G instead of an A. They are phosphodiesters, diester. They are twice estered, which is the bond between the phosphate and the sugar is an ester bond. And if you link the same phosphate to the sugar twice, it becomes a diester. Well, phosphodiester ACE destroys that. It converts cyclic GMP back into GMP. This could be cyclic AMP. It could have been cyclic TMP. It doesn't matter which letter it was. It's just that, remember, cyclic AMP and other cyclic nucleotides have been used as signals before. They're secondary signals. Well, when there's plenty of cyclic GMP there, sodium channels are open and your receptor is depolarized. Okay. But when light strikes, what happens is this rhodopsin activates the transducin, which activates the phosphodiesterase, which destroys the cyclic GMP, which closes the sodium channel. So suddenly it hyperpolarizes. This is the membrane potential on the receptor. This does not mean that the signal from the neuron shut off. We have not discussed, and I don't personally know, how this receptor responds to hyperpolarization. It may be that the hyperpolarization causes it to release a whole bunch of neurotransmitter, or it might be that hyperpolarization causes it to stop releasing neurotransmitter. Then we'd have to ask ourselves, okay, what was the effect of the neurotransmitter? Was it hyperpolarizing our neuron that was coming after this receptor? Or was it depolarizing it? Was it sending it towards threshold? Okay, because here's the arrangement. That's the neuron synapsing with your receptor. Here was where we caught the light. Here was where we responded to it. Here is where the action potential is first generated. This thing doesn't have an action potential. It's not a neuron. It has receptor potential. It's graded. Now, the bipolar cells, okay, are receiving the glutamate. Glutamate, in this case, is the neurotransmitter. Okay, and when the glutamate strikes the bipolar cells, we still don't know if it depolarizes or hyperpolarizes them because different cells, different bipolar cells, can have different types of glutamate sensors, some of which open a channel to let in some sodium and some of which close the sodium channels, or maybe open a channel for potassium to hyperpolarize again. So that's how we process this information. In the absence of light, you're releasing a lot of this glutamate. Okay, in the presence of light, the glutamate flow kind of stops, and that's a bit of an inference because sometimes it's the reverse of this. Remember, all options are available. And then based on that glutamate, whatever its effect was on that bipolar cell, something changes, and it either speeds up or slows down the number of action potentials it was generating. Now, I'm not going to spend a lot of time on color vision, but you know all colors are combinations of red, green, and blue, and that a projection TV, all it has to do is project these three colors and mix them, and you will get all the colors you're used to seeing. Well, you instead of rhodopsin, you have photoopsin that in your cones that instead of just binding light or not light, will go for a specific color of light, a red, a green, or a blue. I'll come back over to your retina here. And instead of telling you how bright it is, what they tell you is what the wavelength of light was that struck your eye. So the rods go, ah, it's bright. The cones go, ah, it's bright yellow. They don't really say it's bright. They just say, ah, it's yellow. Your rods say, ah, it's bright. And so your brain says, ah, I'm looking at bright yellow. The only difference between these different things that are sensing the different cones is which pigment do they put in, the one that catches red, catches green, or catches blue, and the fact that your brain knows which one it's connected to. This is just a picture showing you that if something is close to you, the light coming off of it that comes in through your pupil can be coming off at sharp angles and still make it through your pupil. So in order to make all that light from that pinpoint object that's coming off at several different angles, in order to make that light make one picture, your lens has to be rounded so that it bends that light back inward to make the one picture. But if the light is coming from an object far away, any two rays of light that come in and hit your eye, come in through the same pupil, are probably coming in parallel. So you don't want to bend them as far. You want to leave this lens pretty flat so that instead of bending them hard because they were coming up and down, since they were coming in straight, you only want to bend them slightly to strike the same point. You'll discuss this more in lab. Taste. Okay, we're getting into the end here. So there's sweet, there's sour, there's salty, bitter, and there's glutamate, umami. Okay, sweet, duh, it's a sugar molecule that binds to a chemical receptor. Sour is acids. Okay, you have receptors for different flavors of acid. Salty is, of course, just sodium and chloride, but also potassium and some calcium salts. You taste a variety of them. And then there's bitter, which is basic stuff. Bases tend to elicit the bitter response. Umami is the weird one. Umami is the thing that tastes savory. It's the meat flavor. When you taste something that is meaty, what you're tasting is umami. Do not think of this as protein. I can prove to you that protein actually has no taste. You have no protein receptor on your tongue. All you do is you take a steak and cook it until it's really, really tough and brown, and then taste it. And there will be no taste there, and you will say, ah, you destroyed it. You got rid of all the taste. And I'll say, well, how? And you'll say, the steak is dry. And I'll say, okay, well, dunk it in a glass of water. You say, no, that doesn't help. And I ask you, why is it dry? What came out of the steak? And you tell me the juice is cooked out. And I say, the fats. You go, yeah, because the tastiest meats usually have fat worked in through them. Chicken has much less flavor than beef. Part of the reason, chicken has much less fat in it than beef. So there's fewer fats to taste. Glutamate is a product of fats. Okay. So when it comes down to it, it's the sweet, the sour, the salty, the bitter, and the fatty that you're tasting. It's aromatic fats that you get there. And it's not even just lipids, because oils have much less taste. Stick your tongue down in olive oil and realize that there's very little flavor there. Stick it in butter and you taste more. Lick the edge of a steak and you're like, whoa, what was that? Okay, so it's the different fats that have specific flavor combinations. Okay, and so we just arrange them on your tongue, and we have the papillae, which are your taste buds. Okay, the bumps that you see, and deep down inside them are these little holes for different chemical receptors. And as far as we can tell, each taste bud has all 5 of the different chemical receptors in it. and it has 5 different neurons coming out of it, allowing you to detect which sensor was struck. But the idea that there seems to be one part of your tongue that tastes salty and one part of your tongue that tastes sweet doesn't really correlate very well, because your tongue has receptors for everything in all places on it. So if there is a pattern, it's not a repeatable pattern, and each person's tongue is actually a bit different. Smell, okay, it's just a chemical receptor once again. Just like taste. Taste and smell I'm not spending much time on because all it is a chemical receptor, and those are kind of 2107-ish. Same thing. So these are not these aren't the ones that I spend a lot of time on, but I do spend a lot of time on that idea of vision and on hearing. Those are my favorite ones to talk about. And that is the end of our lecture.